Welcome to the next episode of the Microbiology Tube. So today we'll be talking about the gram-negative bacterial cell wall. So you know the bacterial cell wall is a rigid biological layer that produces a definite shape to the bacteria. So actually the 10 to 40% of the dry weight of the bacterial cell is composed of the cell wall. So this bacterial cell wall is essential for the divisions of the bacteria. So let's talk about the differences between the gram-positive and gram-negative bacteria in very brief. So as you can see, the gram-positive bacteria, these are the thick bacteria, these are the thick cell wall. So gram-positive bacteria have the thick cell wall, whereas the gram-negative bacteria have the thin cell wall. So if we calculate the length, so then it will be about 20 to 25 nanometers. So the diameter of this or the thickness of this gram positive bacteria is about 20 to 25 nanometer, whereas in the gram negative it will be the 10 to 15 nanometer in thickness. So the gram negative bacteria is more complex than that of the gram positive bacteria. So you can see here, so gram negative bacteria is composed of this is the outer membrane, then it is composed of the peptidoglycan layer, the green one, and then after there is the periplasmic space, and then after there is the inner membrane, which is called the cell membrane. But in the gram positive bacteria you can see there is only the peptidoglycan layer and there is the cell membrane. So this is the basic differences. But here you can see the gram positive bacteria have the thick peptidoglycan layer, whereas the gram negative bacteria has the thin peptidoglycan layer. So how the gram positive and the gram negative bacteria are differentiated? So gram positive and the gram negative bacteria are differentiated on the basis of the cell wall, as you know, but we can also differentiate it by staining, called as the gram staining. So the gram positive bacteria will retain the color of the crystal violet and it will be the violet in color. So whereas if we consider the gram negative bacteria, then it will take the safranin, so it will retain the safranin it will be the pink in color. So these are the basic differences between the gram positive and the gram negative bacteria. So let's see here. So this is about the peptidoglycan layer. So how the peptidoglycan layer is formed. So I am not talking about the only the cell wall, I am talking about this peptidoglycan layer that is present in the gram positive as well as the gram negative bacteria. So the peptidoglycan layer is composed of the NAM and the NAG. So NAM stands for the N-acetyl-muramic acid. I repeat, NAM stands for the N-acetyl-muramic acid, whereas the NAG stands for the N-acetyl-glucosamine. So these are the NAM and NAG. So they will be linked together by the beta-1,4 glycosidic bond to form the peptidoglycan layer. So NAM and the NAG are linked by the beta-1,4 glycosidic linkage. So not only that, again the NAM and the NAM is linked. So you can see here, so this is the NAM and this is the NAM. So these are both attached by the NAG, where there is the beta hydrosidic bond. But if you see here, this NAM is again that is attached with the another NAM, and there will be the NAG, again there will be the NAM, and this NAM will be attached with this one. So in this way the NAM and the NAM will be attached to each other, but NAG and NAG won't attach with each other. So NAG does not attach with NAG, so it will attach with the either the NAM here or only the NAM will attach with the NAM. So this is how it is linked. So the next is you can see this is the this is the linkage. So as I have already told you, the NAM and the NAM will be linked. So between this linkage, so there is the link that is linked by the amino acid. So NAM and the NAM, so you can see this NAM is linked with this NAM with the amino acid. So L-alanine, D-glutamic acid, diaminopylamic acid, D-alanine, so these are again joined with this L-alanine, D-glutamic acid, then after diaminopyramic acid, and and then after D-alanine. So these are attached with each other. But this is the case of the gram-negative bacteria. So if you see, the linkage between the gram-positive and the gram-negative is different. So we are talking about the linkage of the NAM to NAM in the peptidoglycan layer. So if you see here, in the gram-positive bacterial cell wall, there will be the L-alanine, D-glutamate or glutamic acid, L-lysine, D-alanine. So there won't be the presence of the diaminopyramic acid. So DAP is replaced by here L-lysine. So DAP is replaced by the L-lysine here. And another distinct thing is that in the gram-positive bacteria there is the peptide interbase. So this is the glycerin interbase is formed. So there will be the pentaglycerin. So 1 2 3 4 5. So there will be the pentaglycerin. So this is the chemical compositions of the peptidoglycan in the gram-positive and the gram-negative bacteria. So if you see here, this is the structures of the gram-positive and the gram-negative bacteria. So in the gram-positive bacteria, you can see the peptidoglycan layer is very thick, whereas in the gram-negative bacteria the peptidoglycan layer is very thin, and this space is called the periplasmic space. This space is called the periplasmic space. So in the periplasmic space there will be the peptidoglycan layer. So but if you see here, if you compare between the gram-positive and the gram-negative bacteria, all the components are particularly the same. All the components are same. You can see the cytoplasmic membrane, which is also called the inner membrane. So these are the same. Even the peptidoglycan layer and the peptidoglycan layer are the same. Only the differences is that the peptidoglycan layer of the gram-negative bacteria is thin and the peptidoglycan layer of the gram-positive bacteria is thick. And we have also discussed in brief about the linkage between the NAM and NAM. So only these two are the differences in the peptidoglycan layer. So the major differences lies on this outer membrane. So if you see this outer membrane, so major differences lies in it. So if you see here, so all these composed of all these outer membrane and the inner membrane, these are all composed of the lipid bilayer. So lipid bilayer, one part in the lipid bilayer, the one part is the polarine, so it is the hydrophilic one, and the inner part, which is like the tail, it is the hydrophobic one. So this is the structures of the gram-negative bacterial cell wall. So if you see here, this is in the downward there is the cytoplasmic membrane, then after there is the peptidoglycan layer, which is very thin, then after there is the lipid bilayer. These are the lipid bilayer. So what is the particular thing that is present is the lipopolysaccharides. The lipopolysaccharide is composed of the O polysaccharides, core polysaccharides, and the lipid A. So together there is the lipid and there is the polysaccharide, so it is called the lipopolysaccharide. Together it is called the lipopolysaccharides. And actually this lipopolysaccharides acts as the endotoxin. So whenever the gram-negative bacteria infect us, then that the cell wall is also responsible for creating the toxicity in our body. So next is the porine. So this is the porine. So this is the proteins. So this helps to enter the nucleotides, nutrients into the cell. So there is a specialized channel. So this helps in entering in the nutrients entering the nutrients into the cell. So here is the next protein. This is called the Brown's protein, BRAUNS. So this is called the Brown's protein. These are firmly embedded into the outer membrane. So next to this outer membrane, in the outer membrane there is also the proteins called the lipoproteins. So these are the lipoproteins. So there is also the lipoproteins which is attached into the outer membrane. So if you see here, the gram-negative bacteria is more complex than that of the gram-positive bacteria. So these are more complex than that of the gram-positive bacteria. And if you see here, the diameter is about 8 nanometers. the length, the thickness is about 8 nanometer on diameter of the outer membrane, and this space, periplasmic space, is about 100 angstrom thick. So the periplasmic space is about 100 angstrom thick. So this was all uh, about the gram negative bacterial cell wall. So I think you have known a lot about the gram negative bacteria. So if you are confused in some of the things, please don't forget to comment upon it, and if you really like the video, please don't forget to like the video and subscribe the channel. Thank you so much. Thank you.